Stanford University.
Which allows to monitor the trafficking of cells. Dr. Contact is an associate professor of pediatrics and microbiology and immunology. He's an associate professor also in the Department of Radiology and is the director of the Medical Free Electron Laser Program at Stanford University.
Thanks, Carl. So I was put in this position in the program today because we talk about, I'm going to talk about immune cell trafficking patterns as it relates to recognition of minimal residual disease, which is by definition comprised of cancer stem cells or tumor-initiating cells. In addition, this is a nice segue into the next session will be largely on immune cell trafficking and cell therapies. So we got into imaging because we had some questions that we couldn't answer by the conventional methods. We wanted to try and develop the tools that would allow us to watch what was going on inside the living body. I like this movie because it demonstrates some of the key principles of imaging, and I'll spend the first 5 or so slides talking about the tools of imaging using bioluminescence and optics. What I like about this movie is it shows you that you have now access to new information that you didn't have before. What we're looking at here is a transfection of a mouse liver with a reporter gene, asking how long does this gene express, to what level does it express, and so we're looking over a 6-day time course at 2-hour intervals and then 4-hour intervals, and demonstrating that we can monitor gene expression in living animals. So now we have access to new information because I'm looking at how a gene is expressed from a liver, using a liver-specific promoter in the context of the liver as opposed to hepatocytes in a dish. We get additional data for these studies because now we're looking at the entire animal. We're asking, did I miss my target? Is this gene expressed in the lung or someplace else? And so I can make some sense of the gene delivery tools. I can look at temporal changes, again, over a 6-day time course here. And if we have an imaging tool that really is easy to use, we can look with fairly fine time resolution. In this case, we're looking at hours, but you can also look at minutes, seconds, and now with some of the new instruments, milliseconds of how things change in living animals. In addition, now the statistics are better because I have built-in internal controls. I can tell you what this animal looked like 2 hours ago, 4 hours ago, 6 hours ago. I don't need to do separate groups of animals. And so I can look at change in individual groups of animals over time. I can use these images now to guide me to specific sites in the body at specific times to sample the tissue, so I can take the correct tissue at the right time, as opposed to sampling a black box, hoping I find the right tissue and sample at the right times. And we can use this now for image-guided genomics or image-guided omics because I know the context of how this gene or this cell is behaving in the living animal. And so we've done a number of studies looking and demonstrating that this gives us more information than we had previously. But I'm going to focus today on immune cell trafficking. But to do that, you need to understand optical imaging or the use of light in living tissues and what you really need to know to make sense of this data. You'll probably see all week long that people are using light to image biological processes without showing you the method or the essential elements of these methods. So what you've been seeing in that little movie in the previous slide is the use of this enzyme, which is derived from a firefly. It requires cofactors such as ATP and luciferin, as well as magnesium and oxygen. Luciferin is this molecule here, and if you inject this molecule into mice where all the cells in the body express luciferase, you can see that there are different patterns of distribution, whether you give an IP injection or IV injection. So when you think about getting this small molecule to this enzyme in every cell in the body, it's the same problem we have with drug delivery, but here you're actually watching real-time pharmacokinetics. And when we looked at these images initially, said this is great. Now we could use luciferin as a model small molecule. We could deliver it in nanoparticles, we could deliver it in liposomes, we could actually refine our drug delivery tools using reporter mice where everywhere that this small molecule or model drug goes, you get signals. Now you